Molecular genetics is the study of how DNA stores and transmits genetic information, and how that information is expressed phenotypically. To do this, we need to look at the molecular properties of DNA and its interactions with proteins. It's useful to be aware of some of the contributions scientists have provided over the last 100 years. The material of heredity was understood to be found in the nucleus of cells. Thomas Hunt Morgan's experiments on fruit flies indicated that genes were found on the chromosomes. Chemical analysis showed that chromosomes were made up of DNA and protein. It was also understood that many of the different arrangements of amino acids, the building blocks of protein, numbered far higher than the different arrangements of nucleotides, the building blocks of DNA. It was assumed, therefore, that the phenotypic diversity from one person to the next, or from one organism to the next, was due to the many combinations afforded by the sequencing of amino acids in protein. Experiments performed on mice by Frederick Griffith, an English medical officer, produced the transforming principle. Mice injected with disease-causing bacteria, not surprisingly, died. A non-lethal strain of the bacteria didn't kill the mouse. These were the controls for his experiment. Heat-killed bacteria of the lethal strain were injected and the mouse lived. Proteins are known to denature and become destroyed when heat is applied, and so far the disease-causing component of the bacteria is still assumed to be a protein. But add the non-lethal strain to the heat-killed lethal strain, and the mouse died. So if the heat destroyed the protein, it can't be protein causing the lethal aspect of the bacteria. Somehow the non-lethal strain was transformed into a lethal strain due to something not killed by the heat in the lethal bacteria. Avery, McLeod, and McCarty, 3 American scientists, took over Griffith's work when he was killed during the Second World War. Verifying Griffith's work, they thought the DNA merely activated gene-carrying proteins. In 1952, scientists Hershey and Chase radio-labeled DNA in viruses. Viruses have protein coats and DNA interiors. Allowing these radio-labeled viruses to infect a bacteria, they saw the radio-labeled DNA enter the bacteria and affecting the genetic disposition of the bacteria. In 1952, scientists Hershey and Chase radio-labeled DNA and protein in viruses. Viruses have protein coats and DNA interiors. Allowing these radio-labeled viruses to infect a bacteria, they saw the radio-labeled DNA entering the bacteria and affecting the genetic disposition of the bacteria. The components that made up the DNA has been elucidated since the turn of the 20th century, repeating subunits of one of 4 different nucleotides. Nucleotides are identified by a phosphate molecule attached to a deoxyribote sugar molecule and a nitrogen base. The 4 different nucleotides are identified by their nitrogen bases. They are thymine, guanine, cytosine, and adenine. A biochemist, Erwin Chargaff, found that these nucleotides were not found in DNA in equal amounts. Instead, he noticed that the amount of thymine was not necessarily found in the same amount as guanine, but there was an identical amount of adenine, and the amount of guanine was matched by an identical amount of cytosine. This became known as Chargaff's rule. The structure of DNA wasn't elucidated until the mid-20th century by Watson and Crick, thanks to the X-ray crystallography of a DNA molecule taken by Rosalind Franklin and brought to them by a 4th scientist, Maurice Wilkins. Watson, Crick, and Wilkins won the Nobel Prize for their work several years after Franklin had died from ovarian cancer. It was found that the crystallized form of DNA was like a twisted ladder, a double helix. One rail of the ladder was a combination of nucleotides, the other rail was made up of complementary nucleotides. The orientation of the 2 rails were opposite, called antiparallel, and are connected at the nitrogen bases. Specifically, the thymine of one nucleotide is always connected to the adenine nitrogen base. and the guanine is always connected to the cytosine. When nitrogen bases pair with specific nitrogen bases in this way, it's called complementarity and explains Chargaff's rule. DNA stands for deoxyribonucleic acid, from a small group of chemically weak acids called nucleic acids. They're made up of nucleotide monomers. Monomers are building blocks for polymers. Nucleic acids are polymers. The DNA monomer is a sugar molecule, a phosphate molecule, and a nitrogen base. The only thing that distinguishes one from the other three. Adenine pairs with thymine, and cytosine pairs with guanine. The 4 nucleotides are arranged in varying sequences along the length of the nucleic acid and paired with complementary nucleotides forming the length of a second nucleic acid. The 2 chains of nucleotides twist to form a double helix.